My name is Marlaina Horewitz, and my advisor is Professor Cotten in the Applied Science Department here at William & Mary, and this is selective binding of Piscidins in 3-to-1 POPE-POPG. Host defense peptides are an integral part of the immune system mechanisms. Piscidins, in particular, come from fish and are able to neutralize cancer cells, bacteria, and viruses. The primary mechanism of action, according to current research, of P1 or Piscidin 1 is lysis, and the mechanism of action of P3 is genetic alteration to cause cell death. In this project, I began initial studies on both Piscidins in an environment that mimics gram-negative bacteria. Using nuclear magnetic resonance, I studied the changes in the chemical environment of oriented lipid samples that mimic the bacteria as peptides were added at varying concentrations. I then compared this to a previous paper published by the Cotten lab group in which these peptides underwent the same testing in an oriented sample which mimics the composition of mammalian cells. A 300 MHz solid-state NMR magnet and a black flox flat-coiled probe were utilized to run P31 NMR experiments. These are the specific parameters of the NMR experiments run this summer and a visual representation of the pulse sequence. Pulse programs dictate the length and power of the applied magnetic pulse. There were 3 types of samples made: a blank where the lipid alone was studied, apostate samples where the peptide was added to the lipid without a ligand, and hollow state samples where the peptide and nickel were added to the lipid. In nature, there is metal available for the peptide to bind to when it is exposed to bacteria. This is a graph of all the experiments run, averaged out, and overlaid on top of one another. This chart quantizes the change in chemical environment when each peptide at each concentration is added to the lipid via the changes in chemical shift compared to the blank. The PE and PG peaks, which correspond to the 2 different lipid head groups, are different chemical environments, so they appear as unique peaks, and the changes in each peak can be evaluated separately. This is key when comparing to the BBA paper and creating a source of differentiation between the 2 peptides. These 2 charts separate the quantization by the specific peptide. The direction of the shifts as compared to the blank were unaffected by changing the concentration of Piscidin 1. The magnitude of the shift was larger for the 1 to 40 samples. This makes sense. As more peptide is added to the lipid, it should have a greater effect. The metallated PE peaks and PG peaks shifted downfield compared to the peaks of the unmetallated sample. Compared to the lipid blank, all of the metallated peptides had PE peaks which shifted downfield, and all the unmetallated samples had PE peaks which shifted upfield. However, compared to the lipid blank, all the PG peaks for P1 shifted downfield, but ApoP3 shifted upfield. This may provide an area of which to test the hypothesis about whether the mutant I2F F19I behaves more like P1 or P3, as metallation seemingly affects the 2 peptides differently. The common PG peak in the 2 lipid systems studied behaves consistently. When the change is significant, unmetallated P1 shifts downfield and metallated P1 shifts upfield. When the change is significant, unmetallated P3 shifts upfield at higher concentration and very slightly downfield at lower concentration. Metallated P3 shifts downfield compared to the lipid blank consistently. This data, available for P3 at the lower concentration, supports a similar behavior between the 2 lipid systems. Analyzing the common peak, it can be seen that increasing the concentration of both systems causes the magnitude of the shift to increase, but the direction of the shift to remain constant. The lipid system has a different chemical shift in the BBA paper for the blank. However, this does make sense, as generally the lipid system was different, and that will affect the PG peak. Moving forward, the P3 series and the I2F F19I series will be completed and compared to the BBA paper. This mutation moves a motif found in both P1 and P3 to the position it is found in P1 on a P3 peptide. The hope is that this is the binding motif. Since P3 fully internalizes into the cell to affect its DNA, and P1 sits on the surface affecting the membrane, if it is likely the binding motif, the peptide will begin to act more like P1. Since these peptides are able to selectively bind to pathogens and not to host cells, understanding the binding motif would allow for further applications of these types of peptides into more targeted mechanisms of fighting disease.